All right, guys, we are on our final day of MJ Biz. You guys still alive?
Oh yeah, I'm here for it.
Yeah, hold on.
Yeah. Good night, though. Good week.
Oh yeah, man. Hawkinson last night. Come
Yeah.
on. Big X the plug. What's up?
Let's go. How was that?
Joe.
That was awesome. Yeah, he came on a little later, but
Yeah,
you wait for the good stuff.
absolutely. Save the best for last. That's right. So I'm here with Jacob and John from Advanced. We're going to be going into their genetic research, what you can expect in 2026, as well as some next-gen nutrient talk as well, kind of that 7th to 8th generation. But let's kick it off. Maybe go into a little bit of your guys' roles with Advanced. I know you guys have a pretty big team and a lot of talent, so let's start with you.
Word. Yeah, so like I said, I said the other day, but Jacob Gibbs, head of U.S. commercial support, started out in the sales team with Advanced Nutrients, came from operations management, running grows, and now I'm doing the commercial support thing, focusing on working with cultivators that have maybe either larger problems than they're really equipped to deal with, or just need a supporting hand for long periods of time, so that way they have somebody to keep calling back, working through things, say, hey, we changed some things, now we're here, what's this look like? Working with the global team, working with R&D, get a little fingers in a few pies, and it's been a good time.
Right on. JJ?
Yeah, my name's John. I go by JJ, the VP of R&D and product development. So we do a lot of work on genomics, discovering new products, and creating new 8th gen and 9th gens in the works right now as well.
Right on. And I want to kick things off with runoff intelligence. So what are you guys doing? I mean, media management for high performance crops. Let's dig into that.
Yeah, for sure. So, I mean, that's the bread and butter of what I do for cultivators. Essentially, runoff is the big key indicator that we can look at that's easy for everybody to look at because you don't have to have any special equipment. All you need to do is run some nutrients through your pot and check what's coming out. The big thing in talking about the runoff intelligence is really what to do with that information. So it's all about trend analysis. You have to start by collecting data in a proper way, so that way the data is useful, and then just analyze what the changes look like going over time. So you're looking at input versus output. We're trying to see what the root zone chemistry is doing. There's a lot of things going on, but it's really simple. And I've found that the industry has made things a little bit more complex in the last 5 or 10 years with the introduction of a lot of the tech that we're seeing, ultimately making it seem as though you can't do all this stuff manually. But we were doing this stuff for years before Arroyo, GrowLink, all these sensors came out, giving people the ability to go a little bit further with their analysis, being able to do that over a larger spread of canopy with the same staff, a little bit more higher level paid staff or something like that. But getting into the runoff analysis, it really is just a matter of trends. So if you're making changes in your cultivation, whether it's with your lighting, with your climate, with your nutrients, you're going to see some of that come out in the back end on the runoff. And if you're not checking this stuff over time, you're not really going to know what's happening in your plant until you see a problem with the plant. And by then it's too late. That's why the problem's there, is because it's been happening over time. And if you're not tracking that, you just don't know what's going on.
Yeah. And let's talk about the trends. I've seen a lot of Coco the last couple years. Let's talk about that. Are you guys seeing Rockwool to Coco as kind of the trend right now going into 2026?
I mean, I would say Coco and Rockwool are definitely the primary media that we see out in the field. A lot of companies that make peat medias have started dipping into the Coco world. There's a lot more offerings of Rockwool out there these days. It's not just Grodan anymore. You got a lot of competition. So we're seeing more competition pushing the small advancements in the growing medium. But Coco and Rockwool are definitely leading the charge for sure.
Yeah. JJ, do you see the same?
Yeah, pretty much the same trends. And also jump back to your previous reply there, with the guys that don't have the systems to build out to do crop steering with technological answers, can you give them some of the brute force ways, like checking your pH, checking your EC, gravimetrics, and kind of explain?
Yeah, for sure. So when we're doing stuff manually, a lot of it has to do with really basic fundamental stuff. So it starts with going back to the growing medium, checking your field capacity, knowing how much water that media is even capable of holding. It doesn't matter what the manufacturer tells you it can. If you're not checking it batch to batch, Coco comes from all the way across the world. Sometimes it's experienced more harsh conditions and it's not going to hold as much water. Maybe you're getting just a bad batch from a new manufacturer. But starting with field capacity, then you can work from there to be able to monitor your performance and your irrigation strategy from that. You can build your irrigation volumes, shot timing, all that stuff by monitoring this information through runoff and dry bag analysis through gravimetric water content. So if you don't have sensors from Arroyo or GrowLink that use volumetric water content, weighing the plant can give you an idea of where you're at in that scale. So if you know where you sit dry and you know where you sit fully saturated, weighing it gives you a really good way to spot check and give some feedback to where you're at in the watering cycle, knowing if you're too far, too short, if you've been chronically underwatering, and it just gives you a way to put together what's going on in that runoff and make that full picture and see it holistically.
Yeah. And I don't know any other nutrient company out there that has generations of nutrients. So I'm actually curious, like, what was Gen 1, JJ? And kind of going into the, generation 7 to 8, let's talk about that.
So Gen 1 would be obviously the original release from Mike. And the way that came out into being was that all the nutrient companies, when they first jumped in, when this thing was in its infancy, had just selected other crops, whether that be tomatoes or peppers, whatever it was. They just took that and tried to apply it to cannabis, but there wasn't really information looking at leaf tissue tests to see what cannabis wanted, at what phases, and how that shifted. So we did a lot of work with BC University up in Canada,
Okay.
a bunch of different strains, and actually looked at what the plant was trying to draw up at different phases, because that does change. Obviously, you got your veg, then you're going to have early bloom, mid bloom, and then you're going to have late bloom. What the plant is demanding at those times drastically shifts, and we didn't feel anybody was actually addressing that. Now, that was before my tenure with Advanced, but they had a team of scientists that did figure that out. So that was Gen 1. And then since then, there's been advancements, incremental advances along the way. And to really announce a whole new generation, what we did is we have a genomics team, we have a nursery, we have a pretty large research team, some chemists, a couple labs. We sat down and went back to square one, looked at literally everything. Let's break this down to its component problems, its issues, its demands, and work forward from there. So develop it from the ground up. So that's what the 8th gen did, is to just really take every single aspect of it, try to compress it, try to address the problem, and address it in a way, or the issue or opportunity, in a way that hasn't been done before. So that would be the 8th gen. That process is realistically 5 years beginning to end to do that. Now, there'll be some discoveries that come along the way, but 5 years beginning to end to actually make a generation. And Advanced Nutrients is the only company I've seen that's actually putting that investment in.
Yeah. It's the only one I've seen as well. I mean, are you guys already planning out like a couple generations beyond? You're just always in R&D thinking about the future?
Yeah, that's my role and my whole team. That's exclusively what we focus on. The thing you have to understand with science is that a lot of it's going to be exploratory. We can look at it. We can look at the microbiology of it. We can look at the microchemistry of it. Theoretically, something might look great, but we have to get there and actually make sure it's going to work in the plant, be feasible, and be something that the market can use. So there's a lot of failures along the way. So we have to look long term and then prioritize, do small tests and then larger scale tests. So it's a process, because these has to go through multiple stages: theoretical, conceptual, beta testing, alpha testing, and then field testing before we can actually put it into a product.
Yeah. And how does the generation impact all your different product lines at once? I know you guys have the new commercial line. What does that look like?
So being a previous cultivator myself, I realized we can't drastically shift everything. So it's going to be an education period where, if you look at a product like our Rhino Skin, great product, legacy product, but with the advancements that came in just silica itself and the way to stabilize it and deliver it. So we're going to be bringing RSA, which will slowly fade out Rhino Skin. Now, we don't want to shock the customers. You've been using Rhino Skin for so long, we can't be like, hey, overnight, it's gone. So a slow phase out on that. And that's going to be the process that you'll see happen with Advanced Nutrients over the next couple of years as we get ready for the next generation. So I don't want any of the growers to be slammed into it. It's going to be a transition. We got guys like Gibbs here. You have the entire sales team where customers can reach out, get that education, to understand that transition.
And I know you guys are turning on Brazil as well as a manufacturing plant down there. When you turn on a new manufacturing facility, are you guys immediately adopting the latest generation technologies?
So that's going to be product by product, and really working with what has the market already done down there. Each market is so drastically different. The way they're growing in Brazil is not the same as America, which isn't the same as Canada or Europe. So that's a conversation that is region and product specific. So I can't give you a blanket answer on that one.
Yeah. No, that makes sense. Well, let's switch gears into genetics. I know you guys have quite a research as well going on over there. What can we expect in 2026?
So with the genetics, what you see in our industry is regional and it's hype, and there's a lot going on. So you have tons of breeders that are out there looking for different attributes, that are looking for different markets, different flavors, whatever it may be. So in order to chase that, what we want to do is we don't want to go after each individual strain that the growers may want. We want to go for the technology that underlines the genetic that they actually need. So let's say you're a grower in wherever, Illinois, Michigan, California, New York, Florida, wherever it may be, you're going to have a different strain that they want for that region. So rather than try to cherry hunt that one for every single region, what we want to create is the underlying technology. What you're going to see in 2026 is our big push into polyploidy. Now, I'm sure people have heard about this already. Well, there's a handful of companies that have released triploids. And a triploid, what it is it has 3 sets of chromosome as compared to the diploid, which is 2 sets of chromosome in normal marijuana. This is not going to be a gene edit. This is not a GMO. This is not something that's unnatural. There's a process by which we can take the plant and we can actually make it increase its number of chromosomes. And for a little bit more background on that, what we do is we take a 2N, which is a diploid, a standard plant. You and I are diploids, plants are marijuana's diploid, all that. We take that and we force it to double itself. And it's not a GMO process or anything like that. And we take it to a 4N. We take that 4N tetraploid, we breed it back to a diploid and make a triploid. The cells are larger, so you're going to have a larger plant. You're going to have a more robust plant. And if you look on those chromosomes, like for example, chromosome 7, in marijuana has 2 THCA synthases on it. So that's what's making the THC. If you look at a hemp plant, same chromosome 7, you're going to have 2 CBDA synthases on it. That's making pure hemp. If you put the two together and breed them, you're going to have 1 THC, 1 CBDA. Those are your plants that make a little bit of both. With our plants, what we're starting to do is, let's say you found a plant you love, you bring it to us, we can make it a triploid. So it's not going to have 3 THCA synthases on it. It's not just the THC. I'm using that as an example. Your terps, your flavonoids, your cannabinoids, and the cell size itself will be larger. So you're going to see a higher yield on it. That's going to be our big push for 2026, and that's the near science. Farther science, if you look at it, there's a lot of really exciting advances that are a little bit more technologically difficult, but that's what we're working on now, where 2026 will be triploids. 2027 may be tetraploids. 2028 may be octoploids. Octoploids is questionable because of the structure of the cell in cannabis. Some cultivars, some plants are amenable to it. Some will just resist it. But that's the work we're doing to discover, because to make nutrients, we have to understand the plant. If you don't understand the plant, how are you going to make the best nutrients? So we invest on both sides of the coin. We're actually look at the genomics, we look at the structure of the plant, and then we look at how to feed that plant.
And is that all under advanced genetics, or where can commercial growers acquire these genetics?
Now, that's a great question. So the R&D entity is actually Tesoro Genetics, and that's a pure R&D entity. It has its own website. That's tesorogenetics.com. That site doesn't sell anything. If you had some research that you wanted to do or you think may partner well with advanced nutrients, that's where you'd go for the pure scientific side of it. From there, the releases of triploids, there's a couple select partners that you'll see come out with it, and we'll let them actually sell it because it's our technology that's going to be applied to their strains. So if you look for triploidy, polyploidy provided by Tesoro Genetics, you'll know you'll have one of those. The other one is internally, we're going to release Trapline Genetics, and the website just went active yesterday.
Traplinegenetics.com.
Traplinegenetics.com, and that's what we're going to use as the vehicle for the in-house genetics we'll release. Right now, there's 4 different triploids that are available there. Come January, there's going to be about 8 more that jump in.
And are you guys looking for licensing deals with breeders out there that want to use like the infrastructure that you guys are researching right now and developing?
Yeah, no, great question. So with that, if there's any of these other producers that want to do a licensing deal, we welcome it. That would be through the Tesoro Genetics website as well. Or if you have your own line, just reach out to either Trapline or Tesoro, depending on what technology you want to license.
Okay. And what are you most looking forward to with these genetics in the field?
I mean, realistically, the consistency of production. So the thing that has gotten people so hyped about triploids initially started in the field, right? The whole idea was that you can have a plant that's not able to be pollinated. It's not going to stop its production of flower to start producing seed. You end up with a bigger, better yield at the end of the year. But once we started seeing that come into the indoor cultivation, we have actually some pictures over on the table of some comparative analysis of a non-triploided diploid versus the triploid in an indoor cultivation, and it's night and day. I mean, one looks like it's never even finished, 3-4 weeks premature, and the other one is double the size. So realistically, the ability to take either the cannabis industry past just the general propagation world, where now you can grow from seed, it was something we had historically seen as kind of unrealistic, not consistent enough. The seed production was not consistent enough. The germination rates, the testing on the seeds, all those things weren't consistent enough to rely on seeds for our general propagation. But once we've gotten to this consistency level for the seed production and the genetics are dialed in, they're in cultivars that we want to see at the shelf, because that's really what it really boils down to. And then cultivators are actually able to take thousands of seeds and run their facility off of seeds, and they're able to get consistent results. There are states that require you to test batches that are the same strain if you've got bags that are purple and green. So the more consistency you can get in the production side of things helps so much further down the line than just growing consistent flower too. So a lot, actually, I'm looking forward to in the commercial space, actually.
Well, we're wrapping up 2025 right now. 2035 predictions. 10 years from now, what will we be smoking? What are the genetics going to look like around the world, JJ?
So here's what's going to happen by 2035, and my guess is, first off, no one's going to be cloning anymore. Cloning will be over.
Cloning will be over.
Yes, there's no reason. Almost no industry actually wants to run from clones. It's a nightmare. You have to have mother space. You have to have all your propagation, and you lose the vigor that actually occurs from seeds. So if you look at the other crops out there, the vast majority don't go from clone, and they only do so when there's some sort of genetic limitation that they cannot get over. Potatoes had that problem for a while.
Strawberries.
Yeah, they're actually solving that in potatoes right now with some gene editing technology mixed with polyploid technology. It's a super interesting thing. It's in its infancy, but that's being applied to potatoes. I'm really looking forward to that being applied in cannabis. With the seeds, you pick up the vigor, you pick up the fact that you don't have to keep the mothers. So by 2035, we'll have moved to that. Also, gene editing is going to be heavily present inside of this field. A lot of misunderstanding in our industry on what TC is, what GE is, and for a quick explanation on that, a gene edit is where you take a gene from that existing strain of plant and you move it to a different plant. It's natural. That gene should be there. It's not some freak thing. That's gene editing. That's just moving it over so we can do that through breeding. It's just the iterations and the time to do so is horrendous. With a gene edit, we can literally just take that technology, chop it out, move it in. That plant's a normal plant. There's nothing wild about that plant. GMO is what people get scared of. And that's where you take foreign or synthetic DNA that does not exist in that species and you jam it into that species. Great example of that is going to be Roundup Ready corn. That gene never existed in a plant before. They jammed it in the corn and then they put chemicals on top of it. And that's really given gene editing a bad name just because of the misnomer on it. So what do I expect? I expect no more cloning. Everything will run from seeds. There'll be light gene editing that occurs on it. And if you look at the very far end of the medicinal side, this would be my wildest expectation, is when you're looking for the whole entourage effect. We know that when you're consuming marijuana, it's not just the cannabinoids, it's not just the terps, it's flavonoids, it's all these other compounds. So that side will remain where it is. If you look at the medical field where they are on one specific compound, they will take that out of marijuana, they will gene edit that and GMO that into yeast and just make yeast produce those compounds. You're gonna see a bifurcation of what we want out of this plant to a purely synthetic chemical branch that some big pharmaceutical company will snag that none of us have an interest in. And then there'll be the actual plant itself from seeds and just vastly improved. And to really kind of put some light on that, if you look at corn, what major crop here in the United States, what corn started originally was maybe the size of my pinky. It was almost inedible. It was hard and it was so tiny. Through selective breeding by humans, we were able to push it to what it is now, which is a massively producing plant. But if you look at how that scale of production occurred, it was a slow, steady for almost 1,000 years until you hit the 1940s. In 1940s, you had scientifically driven selective breeding. You're able to get to true F1s, which no one in marijuana has attained yet, in my opinion. And then from there, you had actual genomic sciences applied, where if you look at it, it's a hockey stick for the production on the corn. That same thing is going to happen in marijuana. The plants will be almost unrecognizable. 10 years would be a little fast for that, but our production yields are going to push up. You want a crazy answer? We're going to get 10 pounds of light by then.
Where I'm sitting is we're still going to be smoking gas and gelato.
So I think one of the things that concerns growers when we're talking about corn, right, is there's a genetic planted in field A. There's a product used to, kill pests in field A. It blows over to field B. Some of those genetics go over there, and now that farmer is sued into oblivion. Do we see any of that in the future with cannabis as far as, patented genetics and solutions?
Definitely going to be patented genetics on it. With corn, what you see is that you're going to have a Roundup ready field next to somebody that has heirloom strains. There's almost no heirloom left in corn. And the nightmare story is the farmer that did have that in the Midwest, and it was pollen, not pesticides, that blew over. So his resulting crop had that gene in it, which is their gene. It's GMO. It's not natural. It's very identifiable in a sequence. In marijuana, when you see these big guys come in, that's where that bifurcation is going to come. It's really hard to imagine that happening in the actual flower side of the industry. Hey, if you look at our triploid production, you can't make seeds from that. So you're not going to have that cross-pollination occur. Now, should some of those larger groups get in, they're probably going to be targeting specific sectors like the medicinal side. So you may have that, but with our crop, we're not releasing pollen. So I don't see that being the biggest issue.
And yeah, go for it.
I was just going to say, I think the area that I see some of these big companies that could potentially step into that space would be taking over some triploid production, where they can now have a secure crop, where they can sell this as a, crop security type of thing to their big farmers, especially once we get to that point where we're seeing 100-acre distillate farms type thing, where we're just doing row cropping. We saw it a little bit with the hemp boom, as far as people doing really big scale cannabis, and hemp is still out there. But once we get to a point where triploids are, cemented into the market, and this is a community-wide thing, I think that would be something where I could see some, trademarked genetics and things in that triploid market where ours are sterile, but we're the only place that has these particular markers. Maybe they've done some gene modifying or something to do some of those things similar to GMO, but yeah, I'd say it's possible. At this point, anything's possible. We're still so early.
Anything's possible. Yeah.
There's a paper that came out several years ago from a very reputable scientist. And what he realized is he could move the THCA synthase gene from, because it's mostly in the flowers, a little bit in the leaves. He was able to unrestrict that, and that gene existed everywhere. The stalks, the roots, everywhere. The entire plant would produce THC. Nobody wanted it. Because the idea for that was, hey, we can use this, we can make this plant when you're going to make some sort of extract. If you want to make a cola or whatever it may be, nobody cares. You just want to get the THC out of there. So that gene modification, that's not an edit, that theoretically existed, and the sequence for it exists, and nobody jumped on it. So you're going to see, what does the market actually want? And that's that bifurcation I said between people that are actually enjoying flower or extracts in their natural, semi-natural state, however you want to call it, versus this hardcore scientific pharmaceutical single or 2 compound hunt.
Will anyone ever own OG Kush, for example, and try to, keep other growers from growing it? Is that ever a future in cannabis, you think?
I think people are going to keep trying. I think that's been something that's already been tried a little bit. Phylos did a big, genetic library for a while, and there was some attempts to try to lock down some things and trademark stuff. But the problem is, it's a living plant. It goes back to people do what they want to do. This was a pirate industry when it started, and people were, very independent. Don't tell me what to do. I'm going to do whatever I want. And I think that there's going to be some of that. There's always going to be some of that. People try to own a piece of the world. But our industry, as in its nature, I think is defiant of that in and of itself.
We did see a big battle with that a few years ago with Philo specifically.
The Gallup.
On a auto rider or a low rider auto flower plant, whatever it was. There was some progeny of low rider allegedly. We did see a big battle with that, and I don't know what all ironed out with that. But one of the things I want to point out is if you are a breeder, if you have a strain that you feel is yours, the best thing you can do is go to Medicinal Genomics right now. It's called Strain Seek. It's like $550. You send in a little sample of your plant, and what they're going to do is they're going to GWS it. There's GWAS. It's the whole sequence. So your whole plant has now been sequenced, and there's a whole bunch of fingerprints in there that will be unique to your plant. So you've now sent it in, you've got it sequenced. They take that and they put it on the blockchain, so it's permanently registered. So should some company, Monsanto or whoever in the future, try to come and say, this is ours, it's not going to fly. So if you have something special, get it sequenced. The best place to do it is Medicinal Genomics.
Hey, you heard it here first. We're actually going to be going out to CanMed, Lake Tahoe 2026. Are you guys going out there?
Yeah, we actually have a package right now. If you are going to, and it'll be on the website in the next few days here, where if you are going to go to CanMed, it's one of the best conventions, in my opinion, in the world. But there's also a deal where if you go, you can pay just a little bit more and you get a whole pile of our 8th gen nutrients, and you get a Strain Seek all for free.
That's a good deal.
That is a very good deal. Go sign up for that now before it's all taken. Well, guys, as we're wrapping up, what are you most looking forward to next year? Could be nutrients, genetics, industry-wide, international markets, Brazil turning on. What are you guys most looking forward to?
So the genetics, for sure. Triploid is becoming a big focus for us, but the 8th gen is also a big priority for us right now, being that we've been teasing on it for the last year and we've been getting everything right, going through registration, which always takes longer than you hope for. And then also, you mentioned the Brazil thing, and it sparked a conversation or an idea in my head from a conversation the other day. We're seeing the passion in some of these emerging markets that we had in this space 25 years ago, where people were just learning about it, they're just eager to get information, and they're really excited. That is what I'm looking forward to, is seeing a bit more of that come on, seeing a bit more of the passion kind of getting stirred back into the mix a little bit, because this industry has been rough for a lot of people, and a lot of people are downtrodden. A lot of people have, got in wide-eyed and bushy-tailed, and now they're a little scruff and gruff, and I think people need a little reprieve from that, but also it's nice when we get to go to some of these other parts of the world and see just that, it may just be in our area that we've been so beaten down. There's still some passionate people out here that are very excited to do whatever they can in their space, and so getting to see a little bit of that is something I'm looking forward to in 2026.
Yeah. How about you, JJ?
So the big exciting things for me, I'm more on the back end, more of the science, the R&D. So really looking further into the actual sequencing. We're getting pretty heavy into that. The increase of the triploid offerings and the increase internally of the tetraploid and octoploid library. 8th gen is kind of out of my hands at this point. From R&D perspective, it's done and launched. So the 9th generation, there's going to be some huge advances. There's some things that we've discovered 3 years ago that we just couldn't get ready in time. So there's going to be some massive shifts in how to feed the plant, the biostimulants, and how to tackle that we haven't seen in any other crop that you'll see in the 9th generation.
There we go. You heard it here first. You have a question up here for these guys?
Thank you. I just wanted to see what advice you had for a tissue culture propagator in this future seed-driven market.
It's a great question.
So if you're going to be doing TC, where the opportunity is going to lie is that with the triploids, there's certain strains that do not like being at that state. We can get them there, but to continue to propagating them out towards seeds, there's going to be select strains. It's ideal to go into TC. So when I say the long term, we're going to get to seeds only, that's long term. In the interim, there's going to be a lot of TC work. Now let's look at When you have a tetraploid, the viability of the gametes is drastically reduced. So to produce seeds from those or even breed with those is a nightmare. That's where my team focuses and really drills in. But to really mass push those out, TC is going to be a critical science in for years, maybe 5 years or more.
We got another question back here.
When you guys are comparing the genetics of other like staple crops, the genomics underlying cannabis traits like terpene profiles and cannabinoid profiles are obviously a lot more complex. Do you foresee that slowing down the genomic selection efforts in this market compared to some of the other markets that you guys referenced?
So there's a handful of groups that have already put out SNP chips, and the development of those SNP chips is a big thing. So identifying the genes of interest and then being able to make a SNP chip is great. There's a group that came out with one a couple of years ago that was interesting, but it just wasn't exact enough. There's another group that's actually here that has a pretty exciting one. It really depends on the trait that you're talking about and what you want to hit. Sex is a multi-allele gene, surprisingly. Everybody thought it was going to be on one, and it's not. Cannabinoid synthesis, very simple. That one, chromosome 7, pretty easy to target. The difference is if you look at the actual sequence of a CBDA synthase versus a THCA synthase, there's overlap. So when you go chop out, delete, whatever it may be, mutagenesis off a THCA synthase, and you only have CBDA synthase, you're still going to get THCA synthase. Same where CBC actually sits in the middle. So that's when you get those bleeders that you'll spit out a little CBC. So it really depends on the trait. If you want to look at terpenes, flavonoids, that is a lot of alleles. That one's going to be a little slower, but what you'll see with gene editing technology, what we'll be able to do just for the research side, which you see happen in the lab is a lot, is we'll be able to pinpoint, let's say, myrcene, for example. We can chop myrcene, and that gives us a really exploratory view on all the other pathways that it's running down. So, no, I don't think it's going to be that hard.
Any other questions back here? Any other questions up here? No? Good to go. Jacob, JJ, thank you for your time today. Appreciate you guys.
Slow, Nick.
Appreciate it, man. Great being on.